This is Roxy. Click on the 3D model on the holotable and let me highlight the relevant objects for you.
Click on the coal power plant. The coal power plant produces large amounts of the greenhouse gas carbon dioxide. Click on the glacier. Now click on the fish farm to your left. It releases large amounts of waste that lead to algal blooms. So our solution is right behind the fish. Now click on the buildings to your right. We are proposing to build large pools. Instead of with excessive amounts of carbon. I don't know very much about. Do you know what?
Select the correct answer below. Option A, the chemical reaction that produces sugars out of carbon dioxide and fertilizers. Option B, the process used by organisms to convert light energy to chemical energy. Option C, the process used by autotrophic organisms to produce energy for all life. No, this is incorrect. Try again. Select the correct answer below. Option A, the exactly. Before
Pigments.
Pick up a lab coat from the rack. Well done. Now go to workbench 1. Find a glove box and click on it. Great! You are now... Click on the ice bucket to open it. Click on the tube pack to open it. Take a 15 milliliter test tube and put it in the tube rack. Choose a fraction for pigment extraction and pipette it into the tube. You need to have a tip on your pipette in order to collect liquid. We automatically pick the tip for you. Have another look at the tube. Are there any pigments in this fraction? Option A. There is something. Choose a fraction for pigment extraction and pipette it into the tube. Do you know where photosynthesis takes place? Within chloroplasts also. Imagine you are a scuba diver the size of a virus exploring huge algae cells. If you want to see how the water molecules are split inside the thylakoid space, how many membranes would you have to cross to get there? Scroll down to see the answer options. You can also click on the View Image button to view. That is not quite right. Imagine you are a scuba diver the size of a virus exploring huge algae cells. If you want to see how the water... That is not quite right. Imagine you are a scuba diver the size of a... Exactly. So, what may extend? Where are the pigment molecules located? Option A, in a protein complex that keeps them in place. Option B, between the fatty acids of the thylakoid membrane. Option C, bound to the phospholipids of the thylakoid membrane. Option D, on the surface of the photosystem. Bravo! The pigment molecule. Why do pigments absorb visible light and other molecules don't? Click on View Image to compare the structures of different pigment molecules and figure out what they have in common. Option A, oxygen and nitrogen atoms. Option B, a metal ion. Option C, double bonds. Option D, extended conjugated systems. Fantastic. In the lab, you must the chloroplasts are suspended in an isolation buffer. To be able to extract the pigments from the chloroplasts, we have to get rid of the buffer first. What's the best way to separate the chloroplasts from the buffer? Option A, use heat to Exactly. Centrifugation is by centrifuge the tube with the orange lid from the tube rack that contains the chloroplast suspension. Wait a second. You cannot run the centrifuge like this. What is wrong with the centrifuge setup? Option A. Exactly. Never forget to balance 2 tubes of the same weight. You should not use a tube filled with water as the weight of it will not match the chloroplast solution. We will use this. Balance the centrifuge and press start. Raise the lab pad for detail instruction. Start the centrifuge. The centrifuge will run for 10 minutes now. In a real lab, you would probably start counting sheep around now. In the virtual lab, we can just 1. Take the tubes out of the centrifuge and place them on the tube rack. Get rid of the buffer from both tubes. The centrifugation has moved the algae cells to the bottom of the tubes. Now you can easily get rid of the buffer floating on top of the chloroplasts. Use your pipette to gently remove the supernatant. You picked up the supernatant. To get a high pigment yield, wear the safety goggles. Please drop the item you have in your hand. Wear the cryo gloves. Shock freeze the samples in the liquid. Close the nitrogen tank lid and wait until the chloroplasts are thoroughly frozen. As this is a virtual lab, I have sped up time and the chloroplasts are now frozen. Open the nitrogen tank and move the test tubes back to the tube rack. Perfect. To elute all the pigments contained in the chloroplasts, what do you have to consider when choosing from different solvents? Option A, an apolar solvent dissolves all the pigments. Option B, water will suffice because the cytoplasm mainly consists of water. You got it. Pigments are organic. We are about to dissolve the chloroplast pigments in hexane and water. Hexane is a very hydrophobic solvent. Hence, it will elute the hydrophobic. Good thinking. Take off the cryo gloves. Add water to one tube and hexane to the other. Vortex both tubes. Great job. Centrifuge both tubes at full speed. Place the two different extracts on the tube rack and have a good look at them. Do your results confirm the initial hypothesis that the red pigment is hydrophobic? View the results again by using the View Image button. Option A, no, the unknown pigment is water-soluble. Option B, yes, because the unknown pigment is hexane-soluble. Option C, no, the unknown pigment is hexane-soluble. Option D, yes, because the unknown pigment is water-soluble. No, this is incorrect. Try again. Do your results confirm the initial hypothesis that the... No, this is incorrect. Try again. Do your results confirm the initial hypothesis? No, this is incorrect. Try... Do your results confirm the initial hypothesis? Exactly. Now, pipette the two extracts into a cuvette each. Nothing to pick up or drop. You added the hexane extract to the cuvette. Take the cuvette rack to workbench 2. You added the water extract to the cuvette. Go to workbench 2. Place the cuvette rack on workbench 2. The blank cuvette. Use the cuvette with hexane as a blank. Interaction is not possible with the item in hand. Great. Now the spectrophotometer. Measure the absorbance spectrum of the hexane extract. Awesome. This is the absorbance spectrum for the hexane extract. Peaks on the graph correspond. Which color is not absorbed by this pigment? Click the View Image button to view a large... You got it. There is a... What kind of pigment is this? Click the View Image button to view a larger image and scroll... Exactly. Use the cuvette that only contains water as a blank. Measure the spectrum for the water extract. Perfect! This is the absorbance spectrum for the water extract. What color of light does this pigment absorb? Click the View Image button to view a larger image and scroll down to see all 4 answer options. Option A, purple. Option B. No, this is incorrect. Try it. What color of light does this pigment absorb? Click the View Image button. Yes. Our hypothesis. Let's meet Roxy to present.
Hey, great to see you again. Did you find something interesting? Option A. Yes, I observed. Well, okay, what else did you find something interesting? Option A. Yes, water and hexane extracts have different colors. Option B. Yes, I observed 2 absorption peaks for... Oh wow, that's great.
Congratulations. If you would like... Let me ask you one last question. Do you feel like you gained...